Thank you for joining us today to learn about our professional assay services team here at ACD and how we can help you move your research programs forward using our expertise in RNAscope technology. I will take some time in the next few slides to go over the basis of our RNAscope technology for those of you that are new to our assays. Our technology is a pioneer in the field of spatial biology. As of recent, we have reached a milestone of 50,000 unique pre-designed probes that are available in our catalog, which can be mixed and matched as necessary for your study. You can even add a custom design for any proprietary sequence or novel target. All our probes do work under universal conditions, so that the same probe can be used across different tissue types and preparations, and for the most part can be multiplexed with other targets as well. RNAscope enables researchers to obtain transcriptomic and spatial mapping information for a variety of RNA targets in both cells and intact tissues. RNAscope facilitates examination of multiple targets at a time in a variety of tissues, using either the fluorescent or the chromogenic readout, while also maintaining a single molecule and single cell resolution. So in short, it is a highly versatile tool for molecular biology research. The RNAscope technology is an ideal spatial analysis solution to interrogate complex tissues. It is a highly specific and sensitive method to detect RNA biomarkers in cells and tissues with a morphological context at the single cell level. The technology basically consists of three parts: a unique target probe that ACD designs against the sequence of interest using our ZZ probe design, a signal amplification system that generates a highly signal-to-noise ratio with a simultaneous signal amplification and background suppression methodology. And lastly, visualization of single-cell RNA molecules in the form of Pontic dots through either fluorescent or chromogenic detection. The assay can be performed on a variety of tissue samples, including FFPE tissues, fresh frozen or fixed frozen tissues, as well as PBNCs and culture cells, and it can be used for simultaneous detection of multiple targets. The selectivity and specificity of the RNAscope technology relies in our probe design strategy. Our probe design relies on two C oligonucleotides to generate the signal. Each oligonucleotide in the RNAscope probe is defined as a single C. The base of the C binds to the target specifically by complementary base pairing. The target-specific binding site is linked to the preamplifier binding site through a spacer or linker region. The top of the C contains a preamplifier binding site upon which an amplification tree is built to aid in the visualization of the target RNA. Once the CC pairs bind to the target sequence, the preamplifier binds to the top of the CC pairs, and to these binds the amplifiers. Each amplifier can further bind multiple label probes to assemble a branching complex at each EZZ binding site. Label probes can contain either a chromogenic enzyme or a fluorophore to generate a signal detectable under a standard bright field microscope or a fluorescent microscope. The amplification strategy allows for visualization of target RNAs as a single dot, where each dot represents an individual RNA molecule. The specificity of our assay relies on this CC pair. If only one of the Cs would bind to the target sequence, the amplification tree would not be able to form, and the single C molecule would be just washed out in one of the stringency wash steps. Our technology has a few options in terms of assay types depending on the target of interest and the needs of the study. The RNAscope assay can be utilized to detect targets that are at least 300 nucleotides in length. This assay can be performed as a singleplex or multiplex assay for the detection of up to 48 targets using our high-plex assay. The BaseScope assay, on the other hand, is designed for shorter sequences between 50 and 300 nucleotides, while the microRNAscope assay is specially designed for very short sequences such as microRNAs, ASOs, and siRNAs, which are typically around 17 to 50 nucleotides long. Lastly, the RNAscope Plus assay combines the detection of any RNA and small RNA targets. All of these assays can be combined with protein detection. As such, our RNAscope multi-omic capabilities combining RNA and protein detection enables researchers to use our technology for multiple applications, such as correlate RNA and protein expression, examine cell type-specific gene expression, validate new antibodies, visualize cellular localization of RNA targets, identify the cellular source of secreted proteins, and many more, as you can see on this list. For the rest of my presentation, I will be talking about how the RNAscope technology can be incorporated into the drug discovery process and provide you with some case studies. Here at ACD, we are aware that the drug discovery process involves many different stages, each of which provides important data that informs the progress into the next stage. The remaining of this webinar, we will focus on how RNAscope technology can be incorporated into each of these stages to advance the research program. I have bolded those steps for which RNAscope and our service team can be a great tool to use. As you can see, except for the final regulatory submission and post-marketing stages, we do have something to offer at every stage of the process. We can help with target identification and validation, lead compound identification and optimization, biodistribution studies once you move into the preclinical study stage, and all the way to testing clinical samples for clinical assay development. At the early stages of drug discovery and target validation, RNAscope can be used as a complementary approach to screen for targets that have been identified from databases or other bulk analysis. Our technology can be particularly helpful for gene targets for which reliable antibodies are not available. Assessing RNA expression can help establish expression patterns with cellular resolution in tissues and regions of interest, as well as in different species and disease models. With that in mind, we have worked with multiple customers who have come to us and used RNAscope to address these early discovery questions. If you recall, we have a tissue bank that is available to our service customers, and very often these tissues are used in projects to screen and validate candidate targets based on the disease, the cell type specific expression, and species. On this slide, I will touch on two common questions we can help address, either by leveraging our tissue bank or using your own tissue samples. The first example on the top of the slide addresses the question of establishing the expression pattern of a target of interest in a specific tissue type across a species. For this particular example, we use an RNAscope syncoplex assay to look at the expression of a gene that is involved in fatty acid production. Here, I am showing the comparison of the target expression across multiple liver samples from both human and non-human primates. This can be used to predict cross-species biological differences that can inform experimental design and even toxicology data interpretation at later stages. The second set of images that I am showing on the bottom of the slide relate to using RNAscope technology to identify changes in target expression patterns in the disease state, or even across different stages of a disease. Here, I am showing representative images from a normal and a Crohn's disease tissues from our tissue bank, as these are some of the most common requested tissues within the inflammatory research space. We actually do have multiple disease indications that are available to our customers. One more reason why RNAscope is a powerful tool during early discovery and target validation is that it can be used to evaluate the presence of specific cells within your tissue and reveal their spatial relationship to each other. In this example shown here, we use RNAscope multiplex assay and specific RNAscope probe combinations in cervical cancer tissues to identify and visualize specialized immune cells within the tumor microenvironment. Here on the top panel, you can see detection of NK cells secreting activated chemokines as indicated by the white arrowheads. In the middle panel, the yellow arrows point to detection of CDC1 cells, which is a subset of dendritic cells that presents tumor antigens to the T cells. And finally, the bottom panel shows detection of CD8-positive T cells. Overall, this data speaks to the ability of RNAscope to spatially interrogate the tumor microenvironment and detect the specialized immune cells when assessing cell-specific expression of a target while developing new immunotherapies. Moving on to the preclinical study stage of the drug development process, once select drug candidates are identified, and it's time to conduct pharmacokinetic, pharmacodynamic, and toxicological studies in different species, RNAscope can be incorporated to study the biodistribution of the candidate itself, be it a vector, a transgene, or an oligotherapy, for example. It can also be used to study the effect of the therapeutic on the target itself across various regions of interest, within tissues from different species, as well as after different routes of administration and various time points after treatment. Importantly, RNAscope is also a great tool to determine whether the therapeutic is reaching or having an effect on specific cells or tissues that may lead to adverse toxicological or safety effects. In the next few slides, I will be presenting data from a publication within the gene therapy space to highlight some of the advantages of using RNAscope to assess biodistribution. Gene therapy is a technology that manipulates gene expression or alters the biological properties of specific cells with the aims to either replace or inactivate disease-causing genes or introduce a new or modified gene into the body to treat a disease. There are a variety of delivery systems that are used in gene therapy. And although what I am showing here may be a bit oversimplified, we can for the most part divide these delivery methods into viral and not viral strategies that are designed to either integrate or not integrate into the patient's genome. Our professional assay services team has had first-hand experience working in projects that involved most of these modalities, and by and large, delivery via adeno-associated viruses is one of the most common approaches that we see come through our labs. As with most delivery methods, part of the challenges with using adeno-associated viruses relate to achieving optimal levels and biodistribution of the vector and payload. To this end, optimization efforts during the preclinical study stage include steps to first, determine the best dosage and dosing administration that achieves therapeutically relevant levels of the vector. Second, steps to improve selectivity by careful selection of the vector serotype, as it is known that each serotype differs in its tropism or the type of cells that it infects. And third, steps for capsid optimization to further improve targeting and efficacy of the AAV vector. So how does RNAscope technology and our service team help in addressing these challenges that we just discussed? Basically, we can help you visualize exactly where the AAV vector and transgene are going to inform decisions on the next optimization steps. We can provide single-cell resolution detection and spatial context, as RNAscope assays can show the biodistribution of the vector and transgene within specific tissues and cells after dosing with various optimized vectors. We can offer multiplex and multi-omic detection of the vector and or transgene along with other RNA or protein targets to better define expression patterns within the tissue and cells of interest. And last but not least, we can provide expertise on image analysis and quantification that can allow you to compare the results to other orthogonal data that might be available. So now let me share with you some data from a recent publication that use RNAscope technology to assess AAV distribution across various tissues at multiple time points following local or systemic injection. In this study, the authors evaluated body-wide biodistribution of an AAV vector in the context of the cell and tissue microenvironments. To do so, mice were injected via two routes of administration, either a systemic IV injection or local IM injection into the TA muscle. And multiple tissues, 12 to be exact, were collected at various time points ranging from 2 hours to 24 hours. I recognize that this is a pretty busy slide, so let me walk you through it. The data shown here pertains to the results obtained from IV dosing. On the top panels, we can see an example of the biodistribution of the vector DNA on the left and the vector RNA on the right, in hard tissues that were collected either at 12 weeks or 24 weeks after dosing. The blue arrows are pointing to the nuclear signal, while the yellow arrows are pointing to the cytoplasmic DNA signal. At the bottom left, we can observe a similar example of liver tissues at 5 days, 4 weeks, and 24 weeks after injection, showing the persistence of the vector signal multiple weeks after dosing. Finally, this is a rather small table, but I wanted to show this data as it speaks to the power of RNAscope for integrating the spatial biodistribution patterns of any target. Looking at the data from the local intramuscular dosing, on the top left panel, you can see vector detection in the actual TA muscle 5 days and 4 weeks after injection, where we can see both nuclear as well as episomal signal. The data also show that even after a local IM injection, there is vector signal in multiple other organs, such as the liver, shown here on the top right panel, as well as the neighboring muscle tissues that are shown in the bottom panel. Altogether, this work shows how RNAscope can be used to generate a detailed spatial and cellular map of target biodistribution that can inform next steps in the preclinical stage of the drug development. To conclude my part of the talk, and before I pass the baton over to Farhad to tell you all about our comprehensive data analysis options, I want to end with telling you about how you can use our professional assay services during the clinical development stage. To date, the RNAscope technology has been used to evaluate tissue heterogeneity and to potentially better screen and select patients. The use of RNAscope-ish as a companion diagnostic has been critical to the clinical success of certain drugs. And the professional assay services team has been an integral part in developing assays for clinical applications and enabling companion diagnostic development. The established workflow includes assay development and optimization, followed by demonstration of specificity and sensitivity, stability, robustness, dynamic range, as well as repeatability and precision. This process involves discussion between our internal pathologists, the sponsor, and the CLIA lab of choice, so that written project reports and SOPs can be drafted and transferred to the chosen CLIA lab for cross-validation. The data shown here highlights the collaboration that ACD had with Leap Therapeutics to develop a DKK-1 clinical assay. For this work, we worked with Leap to develop an assay that could be used to prospectively identify patients with high DKK-1 expression in tumor cells for their clinical trials. I will not go into too much detail about this data, as I will now be passing the baton over to Farhad, who I know will be going a little deeper into this data later in the talk, as she walks through the multiple image analysis capabilities within our professional assay services team.
Thank you, Shomara. I will be focusing on various options of exploring the images for analyzing the spatial patterns of your target expression across various tissues, tissue types, and assays. You can choose from a comprehensive set of data analysis options to obtain the answers you need. Here is a more detailed overview of our analysis offerings. Our priority is to provide you the most appropriate data analysis for answering your specific scientific questions. As such, we have a standard analysis outputs that we provide for our projects, but are always willing to discuss and work with you to generate the most valuable data set for your needs. As mentioned in the previous slide that Shomara showed, the first scoring method we offer is the semi-quantitative visual scoring. This is a fast scoring method that shortens the delivery time. The sample is visually scored using a 0 to 4 scoring criteria based on the average number of dots per cell observed across the entire tissue or the region of interest within the stained tissue. It provides an idea of the level of target expression from low to high and provides a range of percentage of cells expressing your target. The second method is the pathologist edge scoring, which is useful for studies where the marker expression pattern is heterogeneous. This method is more quantitative and involves grouping of cells into 5 bins based on the number of dots per cell. Each sample is evaluated for the percentage of cells in each bin and using a weighted formula to generate edge scores ranging from 0 to 400. Alternatively, bin 3 and 4 can be grouped together to calculate the edge score out of 300. This is useful for comparing with the IHC scores, which also range from 0 to 300. For digital image analysis, we use the Halo software from Indica Labs. Halo analysis is done by our team of highly qualified image analysis scientists and provides expression data on a cell-by-cell basis. Halo analysis requires high-quality images, and for that, we use state-of-the-art slide scanners to ensure optimal image quality and a good signal-to-noise ratio. After the image acquisition, Halo analysis can be done on the entire tissue section or defined areas of interest. After the data is generated, it goes through a second round of QC before it is finalized. The data is then delivered as a spreadsheet along with the graphs for easy interpretation. Finally, our in-house pathologists can help with the image and annotations to accommodate your needs. Our pathologists are also available for teleconference for data presentation and review of data with you. Additionally, we also have customized data analysis. In case any of these above mentioned data analysis offerings do not match your analysis needs, we can also customize the data analysis for you. Now we are looking at the visual semi-quantitative scoring. As already mentioned, semi-quantitative scoring is typically the default scoring system that we offer. It is visual scoring performed to assign a single score to a sample using the criteria outlined on the table, based on the predominant staining pattern throughout the entire sample. For pathologist-guided semi-quant scoring, percent positive covers the entire tissue and the ROIs, and a score is assigned based on the predominant staining pattern. The only drawback with the semi-quant scoring is that the heterogeneity is lost. The semi-quantitative scoring can be done for both chromogenic and fluorescent images. It has a 0 to 4 scoring for expression level. It provides an idea of the level of target expression from low to high and a range of percent of cells expressing your target. It's a rapid scoring system. It shortens the turnaround time and speeds delivery of results. Excellent method of screening large number of samples or TMAs for multiple targets. This scoring criteria that you see on your screen is for the RNAscope assay. The scoring criteria is different for BaseScope and microRNAscope assays. Now here's an example of semicon scoring. What I'm showing is a sustained non-human primate retinal tissues to assess the biodistribution of the vector, shown here as a blue signal, and a transgene, which is shown as a red signal, across different retinal layers. As you can see, we were able to show the differential pattern of expression across the layers. Importantly, I'm also showing an example of our capability to score these signals. In this case, we use semi-quantitative scoring approach to provide the client with the vector and the transgene percent positive cells for each region of interest. Although I'm only showing data for 3 layers of the retina that were scored. You can generate data for any number of layers that you want to visualize and compare. Dual positivity for vector and transgene were also provided. Now, this is visual pathologist edge scoring, which is useful for studies where the marker expression pattern is heterogeneous. This method is more quantitative and involves grouping of cells into 5 bins based on the number of dots per cell. Each sample is evaluated for the percentage of cells in each bin, and using a weighted formula and an edge score is calculated, which again ranges from 0 to 400. Edge scoring is particularly useful in clinical studies. The criteria for edge score remains same for both halo analysis and visual scoring by pathologists. Edge scoring provides heterogeneity of marker expression, which is sometimes lost in semicon scoring. Edge scoring can be done in a specific region of interest like the tumor versus stroma or even specific cells of interest. We can sometimes customize the binning criteria depending on the project. So if you want to bin these cells differently, it can be done. But these current criteria work very well with majority of the assays, tissues, and markers. For example, if you have a low expressing marker, we can customize the binning to match the study. Now here's an example of edge scoring. What we want to highlight here is a collaboration that ACD had with Leap Therapeutics to develop a DKK1 clinical assay. The overall goal was to establish whether DKK1 could be used as a predictive biomarker of response to both monotherapy with their novel DKK1 antibody therapeutic as well as in combination with anti-PD1 antibody immune checkpoint inhibitors. The data that I am showing was published in 2021 and nicely summarizes the studies that were conducted to help optimize and validate an RNAscope-ish assay that could be used as a reliable DKK marker expression in gastric cancers. To this end, we started to perform a series of assays for verifying the sensitivity of our probes and establishing the reproductivity and robustness of DKK1 RNAscope assay. The images on top panel show the ability of our DKK1 probe to detect the target in cell lines expressing from low to high levels of DKK1 in comparing with clients' internal RNA-seq and PCR experiment results. As you can see, the RNAscope assay results correlated with the other experimental data. The images on the bottom left show the reproducibility of the assay across different instruments used to stain individual gastric gastroesophageal tumor samples. And then the image on the bottom right shows the actual staining of clinical biopsies with different levels of DKK1 expression, as assessed by the H score assigned to each sample. Altogether, this is a success story of how RNAscope assay is sensitive and robust technology that can help our clients go through the drug discovery journey all the way from target validation to clinical assay development. While we do look at the staining pattern and the reproducibility, it is the final analysis that tells us about the expression pattern. Once reproducibility is established, it is not only important to visually match the images, but you would also need to analyze the images the best way if you don't go for quantitative analysis. And the best way to do is H scoring, which shows that the pattern is replicated. And the most clinical studies, they go through this. Now I will be talking about the image analysis, the quantitative image analysis in detail. To perform digital image analysis, PAS utilizes HALO software, which is provided by the Indica Labs, which reports the expression data on a cell-by-cell basis across the entire tissue sections. PAS data analysis specialists include PhD-level scientists who are image acquisition and analysis experts, and they employ state-of-art slide scanners to ensure optimal image quality and a good signal-to-noise prior to scoring. Analysis can be selectively performed in regions of interest which are defined according to the objectives of the study. The first step is to annotate the regions of interest for analysis or use a classifier to make sure we identify the right regions of interest. Once the annotations are confirmed, we have the images QC'd by a pathologist. Within any given region of interest, we adjust the analysis settings. A real-time tuning window is used to dial the algorithm to segment cell boundaries and to identify RNAscope dots based on user-defined inputs. The analysis is initiated and the data generated. The cells with white have no expression, pinkish have low expression, red have high expression. After the data is generated, it goes through a second round of QC by HALO expert, this time of the analyzed samples and the data, and before it is finalized. This is done by someone who is not involved in the analysis to reduce bias. The data is then delivered as a spreadsheet along with the graphs to ease interpretation. The three main parts of HALO analysis are the nuclear outline detection, cell segmentation, and ISH dot identification. Once we have a tissue, it is important that we are able to see the nuclei clearly. That is, the staining is optimal. The algorithm starts with identifying the nuclear outline. There are a series of steps followed in identifying the nuclei within the ROI. Then the algorithm is tested on a small portion of the tissue to make sure it is working. If it's a chromogenic assay, the detection is done via color deconvolution. If it's a fluorescent assay, we assign channels. Then we use a nuclear size filters because sizes differ between tissues and ROIs. Next is the nuclear segmentation to detect the individual nuclei that are in the clump. We adjust the settings to make sure we are neither over-segmenting nor under-segmenting the nuclei. This is followed by cell segmentation. This is for marking the cytoplasmic boundaries. We set the radius and the perimeter around the nuclear outlines. We QC by applying the settings in a small area, again to make sure that we are neither over-segmenting nor under-segmenting. Then comes the ISH dot identification. It uses the same criteria as the nuclear outline detection, that is the color deconvolution. We QC for oversensitivity or undersensitivity. Once all settings are in place, we test again to make sure we are picking up the nuclei, the cells, and the dot signal accurately. So there is an important question: how do we quantify clusters? For chromogenic, we assign a dot size per area, so algorithm is able to break the cluster based on the assigned size per area. For fluorescent assays, we utilize both the intensity and the dot size to quantify the clusters. Now, showing you a brief video, we are looking at the coronal section of the mouse brain. I would like to walk you through the basic workflow of HALO. These are the basic annotations. Here we are using classifier to define the ROIs to be analyzed. The analysis is initiated and in progress. You are seeing the cell masking of each of these regions being analyzed: thalamus, habenula, hippocampus, and a closer view of the hippocampus, dentate gyrus, cortex, and a closer view of the cortex. This is how we quantify each channel. Not showing all channels here, then we have a spatial plots for each marker in the multiplex assay. And finally, we have the final data as you can see in this image. Now, why do we need image analysis? Quantitative image analysis is useful for various applications like gene therapy, where we can visualize and quantify the biodistribution of AAV vectors or small therapeutic RNAs across various regions of interest. It can also be used for biomarker or target validation for quantifying the biomarker expression across a wide range of cells and tissues, and also to measure the upregulation or downregulation of biomarkers. In neuroscience, it can be used to spatially interrogate neuronal cell subtypes and networks. In oncology, we can characterize the biomarker expression in the context of tumor microenvironment. It's also useful to quantify the change in cellular abundance, spatial localization, and activation, including the cytokine and chemokine expression. If you are looking to classify cellular subtypes within the target tissue, quantitative image analysis can be really useful. We also have additional spatial modules, including the proximity analysis, nearest neighbor analysis, and the tumor infiltration module, which can be used in specific projects to go beyond the usual data exploration. Use MPS image analysis. You rely on RNAscope experts to obtain accurate and reliable results. Accelerate your time to results with end-to-end RNAscope services from slide staining to image analysis. Access multi-omic results from platforms including Halo, QPath, Visiopharm, and a secure web data sharing platforms. Easily analyze a wide range of tissues from simple to highly complex, define region of interest, obtain custom H scores, and heat maps. Our assay services team comes with a plethora of expertise with direct access to RNAscope technology experts who specialize in ISH services and ensure highest quality is achieved by performing optimization to choose the best assay conditions for your samples, while maintaining the speed with an average turnaround time of 4 to 6 weeks. Thank you for joining us today.